I think the biggest advance in cancer research right now is the development of targeted molecules that can inhibit specific processes that are important for the growth and development of cancer cells. Cancer cells are essentially uncontrolled growth of cells in your body. And in order for these cancer cells to grow, they of course need nutrients. Recent research have shown that cancer cells use these nutrients differently than normal cells. And so what we're trying to do is to use drugs to block the way they use these nutrients so that these cancer cells stop growing in an uncontrolled manner. So in the first paper, we're interested in finding a new treatment for acute myeloid leukemia by performing a drug screen where we tested over 100 different types of compounds. We found that NAMT inhibitors are particularly good at killing leukemia stem cells. The mechanism by which these drugs work is by disrupting the metabolism of fatty acids. An important implication of that finding is now that we can combine NAMT inhibitors with other drugs that also inhibit fatty acid metabolism and combine those two drugs to make them work even better. In the second paper, we studied two drugs that are currently approved for the treatment of acute myeloid leukemia. The first one is known as enasidenib, the second one is known as venetoclax.
The idea was to combine two drugs to see if we can increase the benefits of the treatment.
The way we tested that is by developing preclinical models using human cells and then treated the cells with the combination of these two drugs or each drug alone. The clinical implication of that is testing the efficacy of these two drugs in patients with relapsed or refractory AML.
So the importance of being in the research hospital is to have access to primary sample coming from patients and be able to use them to see if our drugs work.
Collaboration played a big role in both of these studies. I want to emphasize the collaborators that we had for the first study, where they helped us in analyzing the metabolites in the cells before and after treatment. And we also had collaborators that helped us do the actual screening, the drug screening studies.
The best day was when we ended the in vivo experiment, because this experiment was on for more than 6 months. So it was a very long experiment, and at the end, when we finished it and we had all the samples, that was a relief.
In the first paper, although we found that NAMT inhibitors are good at killing leukemia stem cells, what we don't know is what the effect of this drug is when given to a patient and what the side effects would be when given at the doses that are required to treat the leukemia. So this is something that we plan to explore in the future. The most exciting aspect of these projects is the ability to translate them from the bench to the bedside. And so, for example, with the second project, we're able to conduct a clinical trial where we combine these two drugs and actually test how well they work in patients.